Welcome to the Next Generation Sequence Analysis Week 8 lecture on structural variant detection. So this is the one and only opportunity for me to discuss with you the rapidly emerging and now becoming a much more mature field of detecting a type of variation that is very different from the SNPs that we've been talking about thus far in the class. So in this lecture, if you don't already know what structural variation is, I'll be discussing a little bit of the background of the biology and the current interest in medical genetics and other areas of biology where structural variation is of great interest. Be talking about briefly about some of the phenotypic outcomes of different types of structural variation, and then go into the methods that people are now using and applying to short read data sets. We'll be talking a little bit about the errors associated with structural variant calling. Unfortunately, similar to SNPs, there are significant pitfalls with various approaches, and I'll be describing that to you, and ultimately at the end of the lecture talking about some alternative strategies and some ways in which we can ensure the highest quality structural variant predictions. So I just wanted to briefly mention that I did have a required reading for you this week. This is a Nature Genetics review on structural variant discovery and genotyping. This is the Alkin et al. Paper that's now on the course site, the Blackboard website, that is. And I'd like for you to download it and pay particular attention to the sections on short read next generation sequence analysis, because there is a couple of sections on historical methods which are important to know, and I'll be touching on some of those later in the lecture. So I just thought I'd introduce a few of slides on the tremendous interest and why we have a tremendous interest in structural variation. And ultimately the reason is because structural variants are ways in which the cell can become dramatically altered in its function with a single mutational event. We think that that's far less likely for a single SNP to dramatically impact cell function. So one way in which structural variants affect cells is through dosage, and so imagine the deletion of part of a chromosome, or one of the maternal or paternal alleles in a region on a chromosome, such as this one here, where a gene called MAPK in a normal sample on chromosome 5, the read depth along chromosome 5 looks like this in a normal sample, where it looks like we're at about 56 megabase position on chromosome 5. But here in breast cancers, there's a common mutation that leads to the deletion of the MAP3K1 gene. Okay, so several of the exons here in MAP3K1 are missing because of this common deletion. So this is a case where this is a normal read depth, and then in a region where you have a heterozygous deletion, you end up with half of the read depth that you would in a normal sample. So this is normal, this is tumor sample. Here's another example here, a different MAP2K gene. Again, here this is the cartoon of the gene structure in the region. So there's a loss of this, most of the exons in this gene, and then the entire deletion here of the MAP2K4 gene. And here's one more. This is a homozygous deletion in a tumor cell where you have, very low coverage compared to the wild type or normal tissue. So the important thing is that these genes frequently map to cellular pathways that influence the tumor biology. And this is one of the ways in which structural variants affect the cell, and they can lead to the loss of tumor suppressors. So the loss of expression of a tumor suppressor, or the gain in expression in the case of other types of structural variation, gains of expression of proto-oncogenes. So the other types of structural variation that are common in various forms of cancer are large-scale translocations and chromosomal events that similarly can alter the expression of a gene. So here is a sort of a cartoon here showing some just a sort of cartoon of what can happen in the case of a translocation, where say a gene moves to a region where it gains a new promoter because it's now moved across the genome, and so this can lead to a change in expression of a gene, which is known in certain types of leukemias. And then, of course, at the breakpoints where these translocation events occur, or an inversion, for an example, would be another case, you can lead to gene fusion products, which all of a sudden have new functions or new expression domains as a consequence of these structural events. So once again, this leads to altered expression. Gain of expression for oncogenes can lead to tumors, or the loss of expression of tumor suppressors. So these are these Circos plots that I've shown you now a few times. Each of the different colored bars on the outside are the chromosomes. It's a common way in which we show that a large-scale event has taken place is by connecting where it originated, where a chunk of chromosome originated, to where it ended up following the translocation. So these are common in cancers and tumor cells, and so when we do matched pair studies, we frequently will sequence at some deep coverage normal cells. We'll then go in and sequence the cancerous cells, and we can compare the differences in the orientations of reads, for example, to help to detect these large-scale events. So it's not just cancer biology where these types of mutations are of interest. This is a study of yeast done by researchers right here at NYU that I'm familiar with, where what they found was that in many independent cell lines, of this type of yeast, that there was a recurrent, duplication, or in some cases multiplication, of a single region here that contained the SOL1 gene in each of these different independently evolved cases. So each of these red bars is an amplification of the region, that is a region that has duplicated perhaps multiple times. And the idea here is that this leads to the overexpression of a gene called SOL1, which is a high-affinity sulfate transporter, which improves the competitive ability and nutrient status, or nutrient ability to sequester nutrients of the mutant lines. And so these gain sort of a competitive growth advantage in nutrient-limiting conditions because they now overexpress a gene that facilitates their acquiring nutrients. So there's many areas of molecular biology and evolutionary biology and medicinal biomedicine and medicinal genetics where these types of mutations are of great interest. And this is a review here, a paper by Fuka et al. It's one of the earlier reviews back in 2006, even before we had the massive short-read sequencing, where the recognition that these type of events could be incredibly important in generating phenotypes began to be recognized. And so you'll recognize here things like deletions, inversions, translocations, all of which can lead to various changes in the phenotype owing to things like a deletion of a gene which now unmasks a recessive allele, so that all of a sudden this is essentially in the hemizygous state, and so this allele now becomes expressed, whereas it was formerly masked by a dominant allele. Here's a case where there's been sort of the loss of or deletion or translocation of an expression domain that leads to a change in a dosage-sensitive gene. Dosage-sensitive being a gene that is susceptible or whose phenotype is altered by a change in expression. And you can go through these, and I would highly recommend this earlier review, especially if you're interested in the biology of how some of these structural variants operate or are thought to affect the phenotype. Now, I wanted to make sure that everyone understands what we mean when we talk about structural variants. They come in several classes. We have copy number variants, which technically duplications also fall under the term copy number variant. So you have duplications, insertions, and deletions are all changes in the copy number of a piece of DNA in the cell. You have translocations or movements of a typically large piece of chromosome from one region of the genome to another, and then you have inversions, which come in multiple different classes, but are just sort of the flipping of typically large regions of a chromosome. So throughout the next few minutes, I'll be telling you a little bit about essentially there's four broad classes of ways in which we use these short read data sets to discover these types of structural variation. So one of them is what we call read depth or coverage-based methods. And so you all are familiar here. We have read depth here on the y-axis, along some chromosome, and all of a sudden, if there's a deletion in a sample genome, the genome that we have sequenced, compared to the reference, when we map the reads back to the reference, the deleted region in the sample genome was never sequenced because it was missing, and therefore you have no read pileups here in the region of the deletion. So this is a figure from another paper that I posted online because this is a good technical resource for those of you who are interested in the technical aspects of using read depth methods to discover copy number variants in particular. This is an example of sort of a study where they actually went in and ground truthed using known copy number variants that were detected by reliable genotyping methods, and then they used read count data here on this axis to show that in known cases where there's 0 copies, you end up with a normalized read depth of 0. When there's 1 copy, you end up with a normalized read count of 1, 2, 3, 4. So this is a proof of principle that they demonstrated here, that after doing appropriate normalization methods, that you get this nice linear effect here of read depth. And so this is the signature that we are looking for in using read depth methods. So just quickly, a thought experiment. If you have in your sample genome a duplicated section of chromosome, and you sequence that duplicated section of chromosome, but the reference genome only has a single copy that is duplicated in the sample genome. When you map the sample genome, you have twice as much DNA mapping to the single copy that's in the reference genome, and that's why we end up getting this twofold effect. So if that's not clear, just replay the video, maybe just listening to that again. If that's not clear, hopefully at this point, these read depth methods should be starting to sink in. So one of the problems with the read count methods are known biases, and there's a couple of really important ones. So certain regions are susceptible to these biases are either under or overrepresented in the read counts. And these differences in regions, this heterogeneity that we've talked about a couple of times in the class, is different from just sort of the random sampling of DNA and the alteration in the pileup depths that we would get simply by a Poisson or a Poisson-like process of sampling the DNA in our library construction and in our sequencing. So these biases, though, ultimately we'd like to, be able to use read depth differences across the genome to discern differences in copy number. However, the biases that I'm talking about in this slide are not due to differences in copy number. They're due to GC content and mapping or alignment biases. So this is an example here of read counts here on this axis in bin regions of the genome that have different GC content. So this is the proportion of the nucleotide bases that are G or C. Okay, so here's 50%. Much of the human genome is, centered around 40 to 50% of GC content. But what you can see is that at both extremes, high GC content and low GC content, or the AT-rich fraction of the genome, you see significant biases in read depth. And the reason for some of these biases is primarily related to, it's now widespread thinking that this is a result of a PCR artifact, where PCR works optimally in regions of the genome that have a near neutral GC content. And so you end up preferentially biasing the PCR in favor of fragments of the genome in your library preparation that have a sort of a neutral or 50-50 GC content. And so regions then that have lower or higher GC content do not get amplified, therefore are less likely to be represented as a cluster on the flow cell. So another source of error is in mappability, or this is alignment biases, where you have certain parts of the genome that tend to be refractory to mapping and other parts that are less refractory to mapping. This might be due to things such as repetitiveness of the genome. And so this ends up, regions of the genome that have low mappability then appear like they're at low copy number when they're in fact not. So I won't go into the details here, but the Magi paper that I've posted on the course site, if you're interested in the details of how some of the read count methods or read depth methods adjust for this factor, you can see already get a tip off here of how they might do it. These are lowest regression curves here, and so you can fit a model to this, and therefore you can remove the effect of GC content on the read count by doing a numerical correction. So again, if you have a look at the paper on the course site, if you'd like to read further about that. Okay. So in the next couple of slides, I'll introduce the read pair methods and how sort of in principle read pair methods work for insertion deletion detection in particular, and then I'll just mention that it's these same read pair methods that you can use to distinguish between translocations and inversions and other of these large structural events simply by the evaluating the position and the orientation of the mapped reads. So here the concept that I want to get across here with insertion deletion detection with read pair methods is the idea that when you construct your sequence library, you have a known distribution of insert sizes, and the reason you have this known distribution is when you take your genomic sample or your sample of genomic DNA, you size fractionate it using sonication or some other method, and when that's completed, you typically will run an agarose gel. You'll extract a band of DNA that's at a particular size, okay, and you fractionate it this way so that you have a known, section of fragment size, or mean fragment size, in this case between 2 and 300 base pairs. Okay, so this is important for the read pair methods, because read pair methods, you have then an expected distance between a pair of reads once they're mapped to the reference genome. So in the event of, sort of like concordance and no difference in length, insertion, deletion status of between the reference and the sample genome, you have this expectation of somewhere between 2 and 300 base pair insert size. However, if there is a deletion in the sample genome relative to the reference, then what you expect is that the reads will map further apart than the basic expectation based upon this distribution. So in this distribution, we would expect like 95% of the reads to map to roughly 1 to 125 base pairs apart. But imagine now that they are mapping to be 500 or so bases apart, and what that is evidence of is a deletion in the sample relative to the reference. So the reference has an inserted allele, the sample has a deleted allele. So taking that rationale and just inverting it, a deletion in the reference relative to the sample, then remember these red reads here are being sequenced in the sample, so if the reference has a deletion, then these two reads will map closer to each other than they do in the sample genome. So this, I encourage you to spend some time thinking about and reconciling these patterns in your mind. But here, this is an example from a Nature paper published a couple of years ago, where they illustrated the use of this read pair approach together with a coverage depth approach in identifying a large deletion. So the green lines here connect pairs of reads from the sample genome and their distance once they map to the reference genome. Okay, and because these reads map much further apart, okay, into the reference genome than they do in the sample genome, indicates that the sample genome has a deleted allele compared to the reference genome. And you can also see then that the read depth here in yellow also drops in this region. So we're looking at a region of 20 bases or so here, 20 kilobases rather, of a large deletion, where we're missing several genes in that region. So that brings me to the third general class of methods that we apply in short-read sequencing to detecting these large-scale structural events. And in the split pair methods, what we have is a situation where we first align our paired end reads. We have to have paired end data for the split pair methods, as we did with the the read pair signatures. We have to have a reference genome. We map the reads to the reference genome. But in this case, what we're looking for are read pairs where one of the reads mapped and the other read did not map. So the read that mapped, we frequently call it the anchor read, and the read that did not map is unmapped after that initial alignment. But the key here is to recognize that in the sample genome, if that read spans a region that's been deleted in the reference genome, then if we were to split that read and map it back, the pieces back, in the event of a perfect deletion, okay, we would expect the read to then be split by the region that was deleted in the sample genome. Okay, so obviously the sample genome, just to wrap your mind around this, that the sample genome, obviously, when you sequence it, the DNA is what it is. There is no deletion there, but it's relative to the reference that we can observe the deletion. Okay, so when we map it back, we split the read, and in the event of this being a perfect deletion, should map back to two locations that then flank that deleted segment of DNA. So the way this is done is by blast. So typically, we will look in close proximity to the anchor read for these split reads, and these are very computationally intensive methods because you have to blast all of these small little pieces. You actually break up you split the read up into many different combination of two small pieces of DNA, and you blast them back against the region that may be up to 100 kilobases or more away from the anchored read in order to try to detect where the deletion may have occurred. And so this method gives you base pair precision in terms of diagnosing where a deletion is, whereas the paired end method that I just described to you it does not give you that base pair precision. So while split pair reads, this gives the case of identification of a deletion in the sample relative to the reference, or another way to say that is an insertion in the reference relative to the sample. This is the reverse scenario. Insertions are much more difficult to detect with these methods because effectively here we're only able to detect insertions that are in the sample relative to the reference but are spanned by the read itself here. And so these tend to be very short insertions that we're able to detect. So this is a figure from the required reading for this week on the Nature Reviews Genetics article on genomic structural variation discovery and genotyping. And essentially here in this figure they go through class by class and discuss how the first three here are the methods that I just mentioned and how the signature that those different methods yield. So I'd like for you to be familiar with these three patterns here. And I just won't talk about it extensively because I think it's fairly obvious, but in many cases we can do de novo assembly. So imagine you have a set of reads that don't map to the reference genome because there's a deletion in the reference genome. So there's a segment of DNA that does not exist in the sample relative to the reference. And in this case what you can do is you can de novo assemble this chunk of DNA and map it and try to identify this segment that's missing from the reference genome. So these other read pair methods here, read pair methods, as I mentioned briefly, the orientations of the reads can speak to the possibility of there being inversions or translocations or other sort of complicated structural events. And I won't try to explain these here because these are things that are probably best just pondered over by yourself to convince yourself that the norm or forward reverse pairing can be disrupted if there's a change in the orientation in the sample genome relative to the reference genome. And there's a sort of a universe of complex situations here that can lead to various sort of read orientations, and some of the software that I'll be describing in a minute looks for these various patterns and tries to predict cases where you have a large number of read pairs that support a particular type of event. And once again, these are a couple of additional examples here of various different types of structural variation and there's sort of predicted signature for these different types of methods. So once again, this is the Alkin paper. I would like for you to read this paper quite carefully, particularly the parts, the next generation sequence inference methods, and also pay some attention to this figure and see if you can rationalize what these various patterns might look like in short read data after you've mapped the reads back to the reference. So just before we move on from these specifics of how these various methods infer these different classes of events, I wanted to mention that in the end, oftentimes these methods are most effectively applied when the different sort of signatures from the different classes of methods are sort of uniformly give the same answer as to the type of event that took place. So here's a case. This is from a early sequencing study of the first 8 Korean genomes, where they detected the deletion of these 2 exons here indicated in blue for this juicy 2GP gene, and the signatures that they detected were here mean coverage depth in blue, okay, and you can notice that you get a drop in coverage, again consistent with the deletion in this particular individual. That is a homozygous deletion, so that neither the maternal or paternal alleles in the sample genome has the section that is found in the reference genome. And notice here the stretching of the paired end reads. Okay, so given some insert size distribution, these pairs of reads mapped further apart in the genome than they would be expected to given the insert size distribution. So again, this is the signature of read pair approach to a deletion. And here's the split read approach, where you have reads that map here on the edge or on this edge, but their mates are split, okay, by this region right here, so they can get base pair position there, then using the split read to detect exactly where that deletion is in the sample relative to the reference. And then finally, oftentimes we can get some sense of what's going on, and we can correct for some of the biases by taking ratios of read depths across samples. So here, if you take, say, in the numerator, this sample genome's coverage depth, and in the denominator, you take an individual that has coverage in this region, you end up getting a drop in the log 2 ratio. So in this case, the ratio itself can be informative. So while I just mentioned that the clearest examples here are cases where all of the different methods yield the same signature, I also wanted to point out that it's not expected for all of these methods to yield the same signature. It's only in very special cases where we might expect that. And this is evidence of this. This is a very careful survey of structural variants in the human genome that found that there's a large areas of where the methods did not intersect. Okay, so only 303 structural variants were predicted by read pair, split read, and read depth methods, where up to 80% of the read depth predicted structural events were not detected by the other methods. So this is not at all unexpected. It doesn't mean that one of these methods is necessarily more susceptible to false positives than others, although that may be true. These are thought to be real structural variants that have been validated, but were detected differently by the different methods. And you can just imagine a thought experiment briefly for why this might be. Imagine a split read method requires that the exact segments of DNA flanking a region that was deleted must be retained in a particular deleted allele. So the regions flanking the deletion must be retained, when we know that frequently the deletions are caused by movements of transposable elements, which actually delete their flanking sequence when they move. And so therefore, we might not expect a read depth method and a split read method to detect the same events, simply because of the biology of how some of these mutations occur.